Hi, year 11s and 12s. Just another video for cellular biology. This time we're looking at magnification and microscope drawings. So this one's a little bit hard to do online, and there will be a follow-up video where we look at actually using a digital microscope, and I'll show you down the slide of the microscope and how we actually go about calculating magnification of that and how we draw those images, etc. But for the purpose of this video, we're just going to go through a few definitions. So if we start off with magnification, now this is one that generally is not done great in tests and exams. Students often calculate, it's a really simple thing to do, but students often find this quite difficult. So calculating magnification of an image or of an actual size of something can be quite difficult. So to calculate how much something's magnified, all you need to do is do the magnification is equal to image size, so you measure it with your ruler, and you divide it by the actual size that you know. So, for example, you might have a scale that says every 3 centimeters is 5 micrometers, and you measured the ruler and it's 15 centimeters. If you do 15 centimeters divided by that scale, it should give you how many times it's been magnified. Where if you wanted to actually, if you knew the magnification, you knew, or you had the image, you could actually calculate how big that object actually is by looking at the actual size equals to image size, so you measure it with your ruler, and you divide by the magnification. That tells you how big the item actually is. It's a very easy process there. The other one, this is less so in tests and exams, more just an actual thing you need to do in biology and science, is when you're using a light microscope to visibly see an object and then you need to draw it. So this is a skill that you should be able to have, and it's a requirement you have that. It's a bit hard to test in an exam and a test, but it is a skill regardless that you need to be leaving the course with. So, first off, when you've got all your image perfectly, you've seen it, you've got the magnification correct, it's clear in the microscope, you can look down it, you can see it, you can identify features. Now you need to draw it. The first thing you need to do is you need to put a title that helps to identify the specimen. So generally, it's just going to be the name of organism, tissue, or cell. You then need to also include a magnification or scale to indicate the relative size of that diagram. So I'd be including both, to be honest, the magnification and scale, so that when you're drawing it, someone could look at your information and actually figure out the actual size based on what you've drawn. You also need to make sure that you're labeling identifiable structures that you can see in the microscope, remembering you're only labeling what you see down the microscope. You're not going to label things that you can't see but you know are there, because that's just basically, that's not what you're seeing down the microscope. Not the idealized version, you're drawing what you actually see. Like I said, there's a very small PowerPoint, and that's that one done.